I end up with that title because I used to interpret alpha oscillations in a very particular way, and that brought me to a confusion in terms of understanding behavior of the participants we studied back then. So I'm not sure how many of you are familiar with the current wisdom or state of the art of alpha activity, but basically it solves the following problem. So in this traffic situation, say, you have to attend to the dog for obviously reasons, but also maintain overtly the road straight ahead. Now, it's been proposed that due to the wiring of our nervous system, brain areas that have to process the more salient output, which will be the dog, are dominated by decreased levels of amplitude of alpha activity. And whereas there is a temporal functional inhibition of the less important item, which is then dominated by increase in alpha amplitude. And basically, we tend to convert this amplitude differences into a color. So reduction is blue, increase is red. And this cartoon basically manifests in a typical topography that we use in our EEG research. Now, this is an interpretation of functional inhibition of cortical circuits, basically routing information coming into the system in a way that solves a behavioral problem that is don't hit the dog and the cyclist. So my take-home message for today is that while this cannot be refuted, there is an alternative view over this that starts with the internal wiring of the brain rather than the input from the road, the dog, and the cyclist. And a more general way, it's a spontaneous rhythm that emerges out of the actions of the individual participants, which are the neurons. And once it emerges, it brings the neurons into coalition that helps to exert oculomotor action, which is then required for visual acuity. So this is all I have to say today, but in order to substantiate this claim, we have to revisit a little bit the parts of it. And starting from top, what we observe in the natural world is a lot of events, but in order to interpret them, we have to set up premises. How do we think they emerge? So my premise, you don't have to accept it, you don't have to agree, but I invite you to accept it just for the duration of the talk, is that everything, the entire morphogenesis is the way it is because of the gravitational pull on this celestial body. And because of the gravitational pull, every organism that needs to survive it needs to also move around. And action is actually the primordial form that gives rise to perception, memory, and behavioral disposition. So put simply, the central nervous system evolved to support the host's action due to the gravitational constant. So what you see here in these cartoons is basically a very similar images. It's the flocking birds or swimming, schooling fish, or dots on a screen that basically represent where we attend at a given stimulus. So the consequence of my premise is that we actually can already now use the same theoretical and mathematical formalization to infer large-scale organizational principles on all these three seemingly detached levels. That is, we basically can predict the past, current, and future organization of those collectives. But now, if that is not just a simple gimmick, but there is some more fundamental rule that gives rise to that, is that actually possible to think about a common denominator here? So I'll start with the smaller case or the simpler case. So this is a simple decision task here, right? So whether I look at left or right or maintain fixation. And I want to convince you in the following slides that the way we do this It's by means of an emergence of electromagnetic wave or field that basically bias the individual degrees of freedom of neurons that come into coalition and then exert action on the downstream ocular muscles. And this is how we actually solve this simple task. So if this is the claim, we first need to ask, so how do neurons actually control eye movements? And due to the gravity premise, one can also it follows from that premise that the default state of the eye is movement. So there's not a single point in time during our existence where the eye is in a stationary position, meaning the eye muscles do not contract at all. So on the left-hand side is a version of something our eye trackers and we consider a fixation during a baseline. But if you zoom in, the action is continued. It's a known phenomenon. It's sometimes termed as fixation or tremor, etc. But the bottom line is there's always an action, and the assumption of a resting position of the eye along the optic axis is basically premature. So if eye muscles do not have any control, say in a state of a full anesthesia or something, then our eyes actually diverge 23 degrees outward, so away from the optic axis. And we know from empirical experience that this is not the case in our daily life. So it is not granted that we have an optic axis alignment, but it requires continuous oculomuscular control. And so Peter Schiller and Michael Steiker made in my view, a fundamental discovery by asking what and how do neurons on the first stage of the control of the eye muscles, how do they operate? So this is abducens nucleus in the brainstem. That's the first stage of neurons. So pictured here at top of neurons that directly innervate the eye muscles. And so the authors ask the following question. So what will happen if we take a microelectrode and penetrate a neuron in the abducens nucleus and stimulate with a rate of 500 hertz and 25 milliampere? What they observed is they termed it rate code. And rate code means the larger the duration of stimulation, so that's 10 milliseconds, 25, 50, 80, 120, the larger the duration of the stimulation, the larger the ocular deviation of the eye in orbit. So a strictly linear position, a linear relationship between the first neural stage that controls eye movements and the actual movement of the eye in orbit. And you might as well change just the rate of the stimulation, but stay within a fixed temporal window of 80 milliseconds. So you basically stimulate with a pulse of 100, 200, and so on hertz. It doesn't matter. So as time and frequency are related phenomena, so the larger the time point of stimulation, the time duration of stimulation, the larger the deviation. And here is the key point. On the very second stage of ocular control by cortex, this rate code changes dramatically. And the change is the following. As long as you are within a temporal window of, say, approximately 100 milliseconds, the size of the saccade, or the angular deviation of the eye in orbit remains the same, independent of where the eye starts. During 25 milliseconds stimulation here, the eye position is here, and this saccade is made. During 50, the eye position happens to be here, and this saccade is made. During 120, the eye position happens to be here, and this saccade is made. But the same saccadic vector is made. So in other words, it's termed by Schiller and Steiker a vector code, but it's fundamentally temporal phenomena on the very second stage of ocular control by cortical circuits. And it's temporal because it does not depend on the frequency of that stimulation. As long as you stay within the temporal boundaries of 100 milliseconds or so. Now, 100 milliseconds are important. That's the take-home message here when you stimulate a neuron by means of microelectrodes. But the second discovery the authors made later on was the following. So they made the same experiment, but now the penetration is on a neuron in the second stage, in the superior colliculus in the superficial layer, because that layer has a direct connection to the retina, whereas all the remainder of the layers in superior colliculus they're related to cortex and do not have connectivity to the retina. So they asked who controls the superior colliculus vector codes, and the answer is large pyramidal neurons residing in V1. The large pyramidal neurons that we happen to believe we measure EEG from, etc. So when the experiment goes like this, you send a stimulus on-off pulse or some light gradient here and measure the response of these neurons in the superficial layers here and also in the deep layers. So, and this is in an initial state. There is no cooling. So the idea here is you cannot ablate the cortex because you cannot perform the experiment again. The alternative is you can cool down V1, and that means you're going to shut it down for a moment and ask what happens when the stimulus is presented again. And the answer is, in the superficial layers, nothing much happens. So they respond because they're related to the retina. But the deep layers neurons in superior colliculus do not respond to the stimulus because the conclusion is they respond via the top-down cortical control by deep layer pyramidal neurons in V1. So this vector coding operation has two features. It follows a 100 millisecond temporal window and is controlled by cortex and not by the world outside. Now, this map is familiar probably to all of you. We have a retinotopy on the level of the retina, so nasal, temporal, superior, inferior. We have the same retinotopy on the level of superior colliculus, where only the first layer is connected to retina, but the remainder is connected to cortex. And then we have this retinotopic organization of the level of the cortex as well. But so if we lack now, the claim is that the neurons have a motor code, a vector code that is going to evoke the same position on the map. The problem here is that a single neuron or single individual more generally in nature is hopeless in inducing change because of at minimum, the noisy environment all neurons are immersed in. So instead, they need to come into coalition. They need to come into cooperation in order to induce a better decision where and how to innervate a muscle and therefore move the eye around. So to summarize interim here, so there's this very intricate, always 100 milliseconds findings on different levels that we didn't happen to connect yet. So the vector code on the level of superior colliculus is a key structure in oculomotor control, follows a 100 millisecond pace, 200 milliseconds, 2 saccades, and so on. The fastest movement that we can produce is around 100 milliseconds, and that's the saccade reaction time, saccade onset. But that happens 100 milliseconds after the burst of superior colliculus neurons or cortical neurons, as Peter Schiller demonstrated for V1, but here in a more recent case from lateral interparietal area in the macaque monkey. So it's the same idea, 100 milliseconds after the initiation of the burst, there is a saccade onset. Deep layers in V1 are also dominated by slower fluctuation of ongoing activity, which happen to have a duty cycle of 100 milliseconds, and we term that alpha oscillations. And this is important because the neurons that happen to control superior colliculus during cooling and after cooling also reside here. And furthermore, all of the evoked literature on evoked brain potentials that has been acquired to date has this shape of a P1 N1, say for the sake of the argument, width of 100 milliseconds. So I want to come to this section of this transient synchrony here in 2 slides. But to summarize for now, so when we lack a microelectrode during our daily engagement with the world, we probably could think of an ongoing electromagnetic wave that emerges out of the actions of the singular neurons with a duty cycle of 100 milliseconds that with the result is that it brings neuronal collectives into coalition, which in turn results in a higher fidelity of oculomotor action, which is then a prerequisite for visual acuity. Otherwise, we don't see much. So the 100 millisecond is key here. And sometimes we study it as a transient synchrony, as I mentioned, in evoked potentials, but it's still related to the 100 millisecond duty cycle I'm talking about. Here's why. So the width of the transient potential is 100 milliseconds, but the distance from the onset of the stimulus to the onset of that window is around 70 milliseconds. This we know from a recent work from Britta and Sarang that happened to use a electrode that is positioned here on the eye, which measures the retinal cell activity, together with a non-invasive imaging in this case, such that one could measure the delay of the activation here on the retina and later on the cortex, which is 70 milliseconds. And if you do the math, you end up with a latency of N170, which is in all visual experiments, it just termed differently. If it's just a visual experiment, it's termed N1 or N2. If it's phase, it's termed N170, but it's everywhere. I will show some cases in a second. So important message here is the 100 milliseconds burst is still here. It just delayed due to the transition delay from the retina to cortex. And it's the cortex that gives down the signal to superior colliculus to initiate the action. So the consequence of this claim here is there will be no eye movement before this 100 milliseconds. And this is indeed the case. That's an example of participants maintaining fixation. On top is the topography of the eye tracker where the eye's gaze position is. They asked during the baseline to maintain fixation. And the red trace is a EEG, and the blue trace is the velocity of the eye. So the velocity is low as they maintain fixation. At time point 0, a stimulus is shown, in this case is a phase, but within the first 170 milliseconds, the position of the eye does not change. Only after this P1N1 complex emerges, the 100 milliseconds window, shortly after, there is a saccadic reaction time away from central fixation, which is the blue color, towards, in that case, the left eye of the actor in the stimulus. So that's for phase. It's the same if you view complex images, such as affective pictures or whatever. Only after 170 milliseconds, you terminate fixation and start exploration. It's the same in a working memory task where you have to maintain fixation and deal with items. And it's also the same when you have to read words on screen. So transient synchrony initiates oculomotor action, or in other words, there's no eye movement possible before the 170 milliseconds. So why is that the case? Here's an example of a patch clamp neuron in mouse barrel cortex, which does not obviously control the eyes, but it controls the whisking behavior, for example. And so what you should appreciate here is this is the spiking threshold, minus 40 millivolt. And every time the potential here, the membrane potential fluctuation touches the threshold, there's a spike initiation. Now, you can basically sort this data into 1 second segments and calculate how many spikes per second there were. And you sort this in high rate and low rate sparks and compute the power spectrum of the fluctuation here on the membrane. And what you basically see is that when the amplitude of the ongoing activity on the membrane is high, the spiking rate is low, and vice versa, when the amplitude is decreased, the spiking rate is high. In other words, there's an ongoing wave that determines the phase of the neural spiking rate. So that has been used in the context of cognition, say by Clemenceau, Jensen, and others. But the idea of duty cycle determined the opportunity window of spiking. But we can borrow this model, but instead of these spikes somehow coding items or some information that is very hard to define, my point is here that the change in amplitude will change the duty cycle. So high amplitude is associated with a narrower duty cycle, which basically recruits very few diligent neurons and eventually results in a way, less space covered on screen. That does not need to be fixation. It will work everywhere. But the bottom line here is that the width of the duty cycle determines the 2D space of exploration by means of eye movements consequent upon the recruitment of neurons. So if that's the model, then we can ask, do we see that in the actual data? And the answer is yes. That's a very complicated slide. But what you should appreciate and understand here, independent of the cognitive context studied, contralateral to the widening of the duty cycle, that is the reduction of alpha power, there is the variation of eye movements and gaze position, independent of the task. In other words, gaze position, oculomotor action, and variation of amplitudes of alpha activity are entangled quantities. So if you know the one, you can predict the other. And this is not just simple left-right dichotomy. It works for entire space we can cover. So this is an example of an experiment where covert attention has been performed. This dot was never on screen, just to illustrate which actual position was asked to commit and maintain to memory. And the entire thing works also in full darkness, so independent of photons input on the retina. So why does this matter? It matters because of this. The common denominator of nearly all experiments we do in cognitive neuroscience start with baseline. The right-hand side here is virtually irrelevant, whether you study working memory, face perception, some objects or decisions left, right, and spatial attention. It's actually irrelevant because all starts with the assumption that this fixation interval is the baseline. Nothing special happens here. And so we subtract or control our task data against this baseline. But my point is, because of default state of the eye's movement, this is the most difficult task for us as observers to solve. And it requires continuous action control. And so in the lack of microelectrodes implanted, that action control is provided by the duty cycle of ongoing alpha activity. So if you submit to this for a moment and accept this explanation, one could ask, okay, so this static images of gaze behavior maintaining fixation can actually be transferred as a function of time. So you segment a slice here and sandwich it along time axis, so you end up by a time and fixation variation map, right? And so if we do that, say, for stimulus that kicks in at 0, we can monitor the amplitude of alpha activity and then maintenance of fixation. And also we have a topography of which position on screen are actually covered. So that gives a answer that indeed participants maintain fixation as they are asked to. But remember, that's variation along a point. It's not a true absolute rest. So they maintain this, but upon stimulus onset, exploration starts. In that case, it's a phase object, as you see here in this map, and the amplitude is reduced, which widens the duty cycle and allows the actual exploration. Now, this is a 5-second temporal scale. Here's an example of 6 minutes temporal scale of the same observation in a different experiment. The experiment actually does not matter. It's just a larger temporal scale. You can also select slices out of this data and basically plot it, overlaid, and there's not much statistic needed to infer a strict relationship. You don't need to know Morse code in order to fulfill the missing parts here in the magenta color. So if I ask you, you probably do something like this. And this is indeed a Morse code, but of a different kind. So that's a large temporal scale of 2 hours of recording of fixation maintenance and alpha amplitude. And those 3 subjects basically listen to stories, and every now and then they're asked to respond to questions related to the story, such that their attendance to the story is checked, which in this case requires reading and moving the eyes around, as here and here and here and so on. And they do so for 10 hours. So that relationship in all 3 subjects is maintained for 10 hours recording. Pictured here only 2, but the entire relationship is maintained for 10 hours. So it's a very entangled relationship in a sense, again, that if you know the one, you can predict the other. Affective neuroscience also embrace this idea that alpha somehow roots excitation and inhibition in cortical circuits, and all of this, so the event-related desynchronization following stimulus onset has been now interpreted as the indicator of the engagement of motivational systems in the brain. Say you look at this high arousing picture and compare versus a lower arousing neutral image, or cup or some neutral object. But all I've said applies also here. So that's the phenomenon that is supposed to reflect a motivational system. So high arousing images, alpha is high, stimulus kicks in, our high arousing images, alpha is reduced, and it's less reduced for neutral ones. And the difference between left and right side gives this alpha event-related desynchronization. But if you happen to check also what the oculomotor behavior tells you, it's the same picture. These phenomena at bottom and at top are basically entangled. You maintain fixation, and once you're asked to perform exploration, then there is a transient synchrony of 100 milliseconds, and you start to explore what you see in the time by fixation maps and the time by frequency maps in an equal way. So finally, it even works if you are asked to foveal read words. So this is the actual scale of the experiment. So 1 degree of visual angle is around this rectangle around the fixation, and participants are capable of reading words from left to right and right to left. And unbeknownst to them, different words will pop up. So you can split the words into whether they're read from left to right or right to left. Confirm on the level of the actual gaze behavior, there is a clear lateralization. It has to be as per definition. This is the actual scale of an example word and how the bias is. So it's not large, but it's consistent. And so eye movements only start after the 170 milliseconds, and slow potentials that have been related to some linguistic features of the stimulus are also lateralized depending on the direction of reading, and so is posterior parietal alpha activity. So also here, the 2 phenomena are entangled. So the reduction of the duty cycle by means of reduction of amplitude of alpha is related to the way how we explore the world outside, in which direction. So, so far, all I've said is not specific to us. It's present in the non-human primate, virtually in the same cortical circuits as in this example, but it's also present in creatures far distant from us, say the honeybee. So in a sense that if I remove the y-axis labels here and also the cartoons, of course, most of my colleagues would speculate that this image and this is basically the same. There is a alpha amplitude, and once the stimulus comes in, there's a reduction and bounces back to baseline again. Except this is not a primate using visual stimulus or evaluating visual stimulus, but it's a honeybee that evaluates an odor stimulus. And the relationship is very, very instant in the way that if you remove the sensory antenna, so that rhythm effectively controls the space a sensory antenna covers in order to infer the odorant. And if you remove or ablate ampute the antenna, you also immediately ablate the rhythm. So that's the same recording site and the same B. So once in an antenna intact and antenna amputed. This is important because it's also the same in humans. So nature solved that for us, unfortunately. So so far, the literature seems to agree that if you're born blind, a congenital blind individuals do not have alpha. Acquired blindness still do because they still have residual eye movements, but it's more or less similar phenomenon as here. So the actual bottom line here is the decision to look left or look right or go light or go dark cannot be solved by a single neuron in the circuit. Instead, neurons have to come into coalition to bring the gaze to a particular direction, and this is achieved or supported by an ongoing rhythm, alpha activity with a duty cycle of around 100 milliseconds. Now, this exact problem is also a problem for different collectives, say here fish schools, that have to navigate the light gradient, so to find the dark space and avoid light space, that is, in order not to be catched by predators, etc. And the discovery here is that similar to a single neuron, a single fish is basically hopeless to find and track the light gradient, which is the y-axis here. So at some point, after an increase of the mass of the collective, there is an exponential increase in the performance in that the performance flips and the task is solved with higher precision. So also here, a collective has to come together and maintain reduced degrees of freedom and solve the task. Now, the principles that drive this collective behavior are obviously attraction, repulsion, and alignment in the case of fish, but those three words are equivalent in the case of neurons. They are just called excitation, inhibition, and synchrony, and also equivalent when on a larger scale of other collectives, which are called approach, avoidance, and trust. But let's stay for neurons and fish for a moment. So there's three principles. So there are two rules actually that we agreed have to be fulfilled in order to measure non-invasively EEG and imaging. And that is parallel alignment and simultaneous activation of large pyramidal neurons. So these two rules allow a current flowing down the dendrites, which is the red color here, and which is associated also with the measurable magnetic field in case you will measure MEG. Turns out that those very same rules also apply for a collective of fish. So during a schooling behavior, parallel alignment and simultaneous activation of tail beats It's a necessary requirement in order to navigate the aquatic space. So if that's so, what will happen if we come up with the same hypothesis of emergence of an electromagnetic field that in turn controls the degrees of freedom of single individuals, which will then require a larger group size? A single or very few would not be able to produce a necessary magnetic field or electromagnetic wave. So this is a standard EEG. Those of you that happen to work with EEG might understand what this is. So that square wave here is probably a saccade and another event, etc. Here is something happens with this channel, so it's a bad channel and another bad channel. But then we see some ongoing oscillations here and there, except this is not a brain, but it's my pet animals navigating their home aquarium. And I happen to position electrodes inside it in a 10-20 system. And so this is important because if you compare the power spectra of these electromagnetic fields, they're virtually similar. So the axes here differ, but the shape of the spectrum, the red one is a human case, the blue one is a fish collective, is effectively the same. So such that if I remove the axis here again, a very high percentage of our colleagues will speculate about the ground truth of these topographies, which is some parietal central case here, and some visual, probably some motor here, etc. But knowing the ground truth, we can now start to broaden our thinking about, what is actually, what do we measure here? And how is actually what the tools that we apply to measure here, how they actually make the concrete prediction for what we see here, giving the knowledge of the ground truth. So all of this goes way down to organisms like this, which is a sludge worm that I'm picturing here. I used to feed to my animals. And I'm picturing it here because this is a clear collective behavior. So this sludge worm, if it's placed into a container, it's initially floating around, but with time, it starts and organizes into a collective and stays so unless it's been eaten by my fish. And so the moment this collective agency emerges, this is also the moment when we start to see electromagnetic wave as indicated in the power spectra here. So overall, we might have misinterpreted what alpha oscillations actually do. And we cannot refute the explanation that are related to cognition. But my point is that they might be related to cognitive constructs and concepts, but via the oculomotor control, which is a necessary requirement for all of us, for all living organisms that rely on the ability to move. And an emergence of that electromagnetic wave out of the actions of the singles through the principles of attraction, repulsion, alignment, or excitation, inhibition, and synchrony. It's the way a control is initiated and sustained that reduces the degrees of freedom. And the price for that is a collective agency and agreement. Thank you very much. I hope I didn't lost you. I might have, though.